She's licking her tail. Nothing dirty. You ready for some good news? Still going. First up, new research has shown that precisely timed electrical stimulation of the brain can counteract memory lapses. A team of neuroscientists from the University of Pennsylvania and the U.S. Department of Defense found that electrical stimulation delivered right as a person's memory is about to fail can improve memory function. But in order for this method to be practical, the team also had to find a way to predict when a memory lapse was about to occur. So that's what they did. By studying the brain activity of neurosurgery patients, the team was able to identify certain biomarkers that indicate successful memory function. From there, they were able to construct a model capable of predicting when a person's memory was about to fail. The team found that electrical stimulation delivered when the memory is working like it's supposed to is disruptive and makes the memory function worse, but when the stimulation is delivered when memory isn't working quite so well, function is significantly improved. This research could eventually lead to new methods of improving everyday memory function, such as more effective treatments for neurological disorders or traumatic brain injuries that lead to impaired memory function. The research is published in the journal Current Biology. Are you all done? Is your tail clean? Good. Next up, an international team has achieved a major breakthrough in the field of optogenetics. Optogenetics, as you may have already deduced, is the manipulation of gene expression using light. It's a new, rapidly developing field, and that development may have just leapt forward thanks to the work of a team of researchers from the University of Colorado, Duke University, and the University of Helsinki. For the first time, the researchers were able to turn particular genes on and off in cultures of mammalian cells as well as living zebrafish using light signals. For now, it's likely this technology will be used primarily in scientific research, but it's possible that this and other optogenetic techniques will be used in the future to treat genetic disorders that affect humans. It could also be useful one day in the field of pharmaceutical manufacturing. The study on this optogenetic method of gene regulation is published in the journal Nucleic Acids Research. And finally, a story about a new application for that quaint old gene editing technique that was first used way back in 2013, CRISPR. CRISPR, a natural mechanism of immune systems that scientists have harnessed as a tool for making extremely precise alterations to genomes, has been repurposed as a diagnostic tool by scientists at Harvard and MIT. The genome editing version of CRISPR uses an enzyme called Cas9, which targets DNA. This diagnostic version uses another protein, Cas13a, which targets RNA. This version of CRISPR has been appropriately named Sherlock, which is an acronym for Specific High Sensitivity Enzymatic Reporter Unlocking, which is a little forced, but come on, they really wanted to call it Sherlock. In the study just published by the Harvard and MIT team, Sherlock was able to detect the presence of the Zika virus in blood and urine samples, identify specific types of bacteria, detect antibiotic resistance, detect the presence of cancer, and even detect the presence of genetic risk factors for heart disease in saliva. Sherlock can be configured to detect any nucleic acid. It could revolutionize both practical medicine and medical research by providing a simple and incredibly sensitive tool for early diagnosis of cancers and infectious diseases, and for monitoring the effectiveness of treatments. The study on Sherlock is published in the journal Science. Precisely timed brain stimulation can counteract memory lapses, light can be used to regulate gene function, and a new form of CRISPR called Sherlock can diagnose and monitor disease like nothing else before. That's the good news. Hey folks, one more thing before I go. I hope you enjoyed this video. If you did, please like, share, and subscribe. And also please consider helping me to make more videos like this one by supporting this channel through Patreon. you can go to patreon.com/Steve Shives to become a patron. Thanks for watching, and we'll see you next time.